this figure. Okay, bonsoir à tous. I will switch to English, of course, since tonight's conference is in English, and it's my pleasure to virtually chair tonight's conference. And as you know, this conference is part of the conference cycle of the DIU MA² course, which is a 2-year program dedicated to physicians working or aiming at working in memory clinics. It's opened to physicians, whatever their original specialty, gerontology, old age psychiatry or neurology. And this course is also available to speech therapists, neuropsychologists, or researchers. And this conference tonight in particular is open to the Lille neuroscience community. So we are now around 70 to be connected, and I'm thrilled to welcome and introduce you tonight, Professor Marilu Gorno Tempini. So you are a behavioral neurologist. You were trained in Italy, where you obtained your MD and specialized in clinical neurology. Then you moved to London, where you obtained a PhD in the University College of London, and you became an expert there in the neuroimaging of language. But you soon moved overseas, and it's been quite some time now that you've been working at the UCSF in San Francisco, which explains why it's early morning for you and late evening for us. You are the director of the Language Neurobiology Laboratory of the UCSF Memory and Aging Center, headed by Professor Bruce Miller, and you co-direct the UCSF Dyslexia Center. So aging, dyslexia, those two keywords emphasize something fascinating about your work, because through the prism of language, you cover aspects of cognitive neuroscience that go from neurodevelopment to neurodegeneration. And even more fascinating is the link that you make between the two in your recent publications that suggest that neurodevelopmental changes might influence susceptibility to neurodegenerative diseases. And something else I appreciate about your work is that as an Italian researcher in the US, you show a lot of interest in the phenotypic differences of aphasia according to the maternal language. And I read with a lot of attention your recent publication about that. You also published recently on the effect of bilingualism on cognitive reserve. But last but not least, I won't skip the reason why every physician working in a memory clinic knows your name, and that is because you are the author of the new 2011 criteria for PPA, primary progressive aphasia, and its variants. Those criteria had the great advantage for us to make a complex topic understandable, and to make this complex topic of PPA accessible for non-aphasiologists. So it's a great honor to have you with us tonight for a conference entitled Language Disorders Across the Lifespan. Marilu Gorno Tempini, if you wish, the floor is yours.
Thank you so much. Thank you for having me. In this past a few months, I've been happy to accept Zoom invitations, but on the condition that then I will have an in-person invitation in the following year, because I miss my travels. I miss meeting people in person and hearing ideas and seeing how different clinics and different researchers work across the world. I think we can all learn so much from each other and from our patients, and how different languages and cultures influence how these disorders that we study present. So thank you for the kind introduction. It has been, in a week, it will be 20 years that I'm in San Francisco. It's incredible. Like you made me think about it in your introduction. I would have never thought that my life would have been away from Europe, but the incredible opportunities that were offered to me at UCSF and by Bruce Miller and the whole team at UCSF had just kept me glued here. But again, I love to travel, so I hope I'll see you soon in person. So what I prepared today, it's a little bit of kind of travel through the history of this disorder that I started studying when I moved to San Francisco after a few years of really basic science of language, and really wanting to apply what I had learned about the network cognitive neuroscience of language to a disease. And I was fortunate enough that when I arrived at the MAC, that at that point was 10 people. Now is more than 300. But back then in 2001, we were only 10 people, and all of those 10 people, the other 9 people were interested really in behavior and not language. So I had the opportunity of taking all the PPA patients and trying to study them through the lenses of really the cognitive neuropsychology and cognitive neuroscience that I had learned in Italy, first with Professor De Renzi and Professor Kappa, and then in London. And the approach that I found in the US was very different, and it was quite striking to me. There were some disorders that, and some symptoms that were analyzed and paid attention to much more in the US than in Europe, and vice versa. So semantic memory was very established in Europe, and it was not in the US. Praxio-speech was a very clear concept in the US and not in Europe back then. And some of the realization came later that maybe this had to do with the differences in the languages that we were studying. You know, the apraxio-speech is more common and symptoms are more apparent in Germanic languages, and maybe difficulties in syntax are more apparent in early symptoms in Romance languages. So with kind of that background, we go back to studying language. I always like to study this picture of Broca's patient, especially to an audience like yours. This is the brain that is stored in Paris that Nina Dronkers had the opportunity of scanning a few years back. So is the brain of Le Borgne that we all know was the study of Le Borgne was the beginning of the cognitive neuroscience and behavioral neurology. And language was the first model we had. So this was the first observation, and it's not that long ago, if you think about it, that a disorder of thinking and producing language actually came from a lesion in the brain. And it's quite striking to think about that this was really the first time that that association was made, that really a lesion in the brain will cause a behavioral change in a patient. As we learned in 2 centuries of research of language, Broca's area is a big area. There's gray matter and white matter. And so the concept of Broca's aphasia has changed over time, but it all started here. It all started here, and my slides are not advancing. Let's see. The fact that language was studied for a long time on stroke patients really influenced what we thought was the network involved in language processing. So language, the common knowledge is language is stored in the perisylvian or processed in the perisylvian cortex. That was classic neurology teaching. And slowly we realized that that was a bias caused by the fact that vascular disorders really hit those areas much more commonly. So it really became apparent that we had to study different diseases, different disorders of the brain to really map cognitive functions, especially when it has to do with language, which is a complex human, typically human function that is distributed across most of the left hemisphere. So in this context, in which aphasia was really caused by vascular, it was known that let's say the dogma at the time was that Aphasia is caused by vascular accidents, and language is stored and processed in the left perisylvian cortex. In the early 1980s, Mesulam had the genius of the first observation of primary progressive aphasia in his clinic with not much resources. You have to think that back then, the reports of this disorder was just his clinical observation at the bedside with really no research scans or nothing like that, maybe a CT scan. And Marcel's observation was that these patients had a progressive disease. They didn't have a stroke. They didn't have an acute onset aphasia. They had a slow onset aphasia, and he gave it about 2 years. So for a while, it was only a progressive language disorder, described basically using the nomenclature and the thought process of stroke aphasia. So in broad sense, it was a word finding, naming, and word comprehension deficits. Isolated and progressive. And this was quite a description, because at that time, dementia was a disorder of memory, and that's it. That's how it was described. So this was really out of the box thinking back then. And without having much evidence on his patients, much, as we said, neuroimaging on these patients, the thought was the atrophy must be perisylvian, because language is stored in the perisylvian cortex. So language, so in these patients, atrophy, it's a neurodegenerative disease, so it causes atrophy. It should be in perisylvian cortex. And also the neuropathology must be non-Alzheimer's, because Alzheimer's disease causes memory problems. It doesn't cause language. And these were all completely plausible hypotheses that the field worked through the years, and some were right, some were wrong, and we'll go through how we evolved over time. So when I started looking at these disorders, again, I started through the lens of the European literature, and then the US more literature. And in Europe, the focus was really on semantic dementia, described in England, first time by Elizabeth Warrington, and then by the group at Cambridge, John Hodges, Caroline Patterson, and really was the description, very detailed and a very cognitive neuroscience approach to what semantic memory is. And we'll go through it, but it was a very distinct, beautiful description of this syndrome. Very first use of voxel-based morphometry in London, where I was doing my PhD, actually not studying these disorders, but sitting right next to the person, to Kath Memory, who published the first VBM studies in semantic dementia. And it was very clear that the atrophy was anterior temporal. And so this didn't really fit with the perisylvian atrophy that Marcel was describing in the US, and there was a little bit of a disconnect, and the field kind of debated this. In the US, they were called progressive aphasia, fluent aphasia, semantic in the Europe, they were called semantic dementia. How do these two disorders interact with each other? Are they the same thing or not? The atrophy seems to be different. So that was the question there in the beginning, late 1990s. At the same time, about the same time in the East Coast of the United States, Murray Grossman and his team were describing another syndrome that was called progressive non-fluent aphasia, PNFA. So many of you who look at this literature might have all these different abbreviations, but it was basically this third syndrome of progressive and non-fluent aphasia, in which the atrophy was described at left hemisphere, but the clinical picture was kind of different, both from Mesulam's PPA and Hodges' semantic dementia. The patients were non-fluent, they had hesitations, but they also had anomia. It wasn't really clear. So this was, to me, how I started in the early 2000s. I think that was kind of the state of the literature. And I kind of stepped back a little bit and took all the PPA patients, defined as broadly as possible, as Marcel Mesulam had described them, and did the first analysis of their atrophy, and showed that if I grouped them all together, there was both frontal parietal, and also anterior temporal atrophy. So it was really the frontal temporal dementia description of anterior temporal and mainly frontal atrophy, but also this parietal region that wasn't quite that expected. And so what then we did is take a really cognitive neuroscience approach and did an evaluation of these patients that looked not just of language symptoms like anomia or word comprehension, but really tried to go through every domain of language. So motor speech, fluency, semantics, comprehension, reading and writing, and basically taking all the aspects that had been described before and test them systematically in all of these patients. And then what came out was the description of really 3 variants, not just 2, not semantic dementia and non-fluent, progressive non-fluent aphasia, but really 3 syndromes, with this 3rd one that we named logopenic variant. And for the young researchers in the audience, this first paper that was published in 2004 really took 3 years. It was 3 years of review and re-review and rejection, and it took a long time. So if you have an idea that you really believe on, stick with it. Don't ever take rejections. And so we ended up with these 3 variants that we will talk about today. They're by no means the only variants. They are the most common variants. The domains, what is really important to think about is that if we have the right tools to study and examine these patients, it is possible to distinguish the anatomical involvement in the brain. And we have to have the right tools, because these are really sometimes early on mild disorders. And some of the tools that were constructed to study stroke patients don't work for these patients. But what is really important is that we need our localization from our clinical evaluations, but also the prediction, the fact that we diagnose these 3 main variants is really important to predict what kind of biological underpinning. So what is happening in the brain to cause this syndrome? Not just the localization, but also the molecular cause. Is this Alzheimer's disease? Is this frontotemporal dementia? So there are really 3 levels of analysis. There is the clinical syndrome, so what the patient presents, what kind of deficits the patient presents, to what anatomy, network anatomy, it corresponds in the brain, and then what is the molecular pathology that causes the deficits? Is it amyloid and tau, like in Alzheimer's disease, or is it only tau, as in a progressive supranuclear palsy or cortical basal degeneration, or is it what we call a TDP pathology in the case of semantic dementia? And while the correspondence between the clinical syndrome and the anatomy is, I would say, with the right tools and the expert clinicians, is almost 100%. The correspondence between the anatomy and the molecules is not perfect, is probabilistic. But dividing these 3 variants, I think, helped us a lot. And we'll go through how. So in our clinical pathological conferences, this is just to try to make you understand the relevance of this. The fact that from a clinical anatomical syndrome, we can predict with some accuracy, not perfectly, but with some accuracy, the pathological causes, it means that language can help us distinguish really between different variants of frontotemporal dementia and Alzheimer's. And it can help us then put patients in the right clinical trial for the right therapies, which in turn will help us treat them appropriately, both symptomatically and for the new therapies that we hope will come out. So the logopenic variant, the new variant that kind of we described, the one that in which atrophy is posterior temporal and parietal, is most often caused by Alzheimer's disease. Not 100%, but I think the literature is pretty consistent now that about 90% of the patients actually have early age of onset Alzheimer's disease. They're usually in their 60s. The patients with the non-fluent variant that we'll see is mainly characterized by a problem with motor speech and grammar, is most often caused by frontotemporal dementia type of pathology and of a tauopathy. The semantic variant PPA, which is the name we give to semantic dementia to keep it in the umbrella of the primary progressive aphasias, is most often caused by TDP pathology. So they're basically 3 different diseases, which makes it hard for me in one lecture to describe all 3 of them, but I'll do my best. This is just kind of an introduction to kind of make clear the significance of how this disorder can really help us understand neurodegenerative diseases in general. And the fact that we have so much cognitive neuroscience of language already to build on made it easier for us to identify variants of this left hemisphere neurodegenerative disorder. The correspondent right hemisphere neurodegenerative disorders that we call more in general frontotemporal dementia or behavioral variant FTD still has a little bit more of a difficulty phenotypically dividing the patients because we just have younger science in behavior and emotion processing, but I think we're making huge progress there too. So why do these degenerative diseases? This is a question that is very dear to me and my colleagues at the MAC. Is a big emphasis of ours. So this is a slide that I borrow from my colleague Bill Seeley. And we together think a lot about network-based vulnerability. What does this mean? I think it means, like we said, that these patients really have Alzheimer's disease. So the patients who have this selective atrophy in the left temporal-parietal cortex in the left hemisphere, and we'll see have trouble mainly with the phonological aspects of language. Actually, when we look at their brain post-mortem, and now also with biomarkers during life for Alzheimer's disease, they have Alzheimer's disease. So why does it present so differently? And why does that area, that network in the brain has vulnerability for this disorder? And why doesn't it present as a run-of-the-mill, usual amnestic Alzheimer's disease? This is a question that we think if we can answer, it will help us a lot understand the biology of these disorders. And so the general model, there is no data in this slides. It's just a model that we like to use. So we think of the brain as divided in epicenters and nodes. This is what the latest neuroscience tell us about the organization of the brain. There is not one single area that does one single function, but there are networks of regions that interact together to produce behavior. So let's think that these are epicenters and nodes. We know that each disease has an epicenter, an area where it prefers to start. And that's why progressive non-fluent aphasia, the variant that was described in red in that brain, starts in frontal lobe, and the variant that is described in blue in the figure starts in temporal-parietal, and why semantic dementia starts in the anterior temporal lobe. That's the epicenter of that disease. How that disease spreads depends on this network anatomy of the brain. And so we can really predict, based on where the atrophy is in the brain and how it spreads over time, it can help us predict what the pathology is, but also the progression and the clinical course of these disorders. But why is it that that disorder it hits this specific epicenter? There must be some intrinsic vulnerability of that region. And we know that in other neurodegenerative diseases, in Parkinson's disease and Huntington disease, there are certain cells, certain areas of the brain where the disease starts. So there is intrinsic vulnerability. What can cause it? Of course, genetic and developmental background. And I should say that the classification of PPA that we used and we thought about was really pre-progranulin discovery, let's say pre-genetics. So it works much better for cases that have non-genetic presentation of the disorders. And this is a little bit more for the experts in the field. But so we have this intrinsic vulnerability can be caused by genetic, and we'll see our hypothesis is also by developmental background of the individual. So how the brain developed, environment, and probably a lifetime of brain usage, in which I'll also give the idea of different languages that are spoken, or multilingualism. So what we call cognitive or neural reserve. So because of all these factors, there is this epicenter one that has higher vulnerability for a certain disorder. So when the disorder starts, for whatever reason that we don't know, it first hits that epicenter. And from that epicenter, it will spread. And we know from basic science now that, for instance, the protein tau spreads transsynaptically. So it spreads from one neuron to the next. And so the neurons must be connected. And so if we can learn about the network connectivity in the brain, we will learn also how these diseases progress. So from that epicenter, the spread will be in the closest, most connected node, and then across the different connection across other networks in the brain. So we have techniques now with which we can measure networks in the brain pretty easily. This is an example of how we use what we call resting state fMRI. So patients or healthy controls can just lie on the MRI scanner for 10 minutes. They don't need to do any tasks. I did my PhD in activation fMRI. It took me a long time to kind of give into the resting state kind of idea, but it's incredible how well it works if we choose the right epicenter. So again, we need to have the basis of cognitive neuroscience. And I'm really, really adamant in saying that, especially to the ones of you who are thinking of going into this field. I strongly believe that the practice of clinical behavioral neurology has to be based in the latest cognitive neuroscience theories and tools. And so we use this fMRI resting state to look at those networks in healthy controls, so that then we can help, we can use these as model for the spreading and epicenters of the diseases. So this is an example. We take 10 minutes in healthy controls, and these are kind of the seeds. We look at these regions and see how they connect to the rest of the brain. And if you do that, you can basically map the whole left hemisphere. So, and based on what we know from the literature of cognitive neuroscience and activation fMRI and patient studies, we can really understand that each of the main language domains are interacting and segregated in the brain, and they basically map the whole left hemisphere. And we can use this as predictions on how the disease is spread in our patients. We also have tractography now, so we can look also not only at functional connectivity, but also at structural connectivity. So white matter connectivity within these same networks that we have defined. So our hypothesis is that the neurodegenerative diseases that we're studying will spread through these predefined functional network for language that can really be identified with the 10 minutes of an fMRI scan at rest. And this tool we're also using now to study recovery. We're using it to study functional network in pre-operative tumor patients. Is really a really powerful tool also, I think, for clinical usage. So these are the structural. And then let's see, 9 o'clock. Okay, I might go a little bit over the 50 minutes. Is that okay? Or do we have a hard stop?
You have all the time you wish.
Oh, don't say that. That's dangerous. I'll keep you there for dinner. No, I'll maybe extra 10 minutes. But so after all this long introduction that I hope gave you a little bit of context on the importance of studying these disorders, so we can go through all three of them, and maybe in the panel discussion we can go in more details on the latest studies. But so the first one is what we call non-fluent progressive aphasia. And again, the work on the criteria in 2011 was basically an agreement to disagree. It was, you know, you can see that from the paper in 2004 to the criteria, there were almost 7 years. It took a long time to kind of get to an agreement of a general nomenclature, and I think it will evolve and it will be fine. Criteria should evolve with the science. And so in the non-fluent progressive aphasia, primary progressive aphasia, there was a description of PNFA as well, and also what now is called primary progressive apraxial speech, which I think should be called primary progressive motor disorder. So basically there are two main symptoms in this disorder, in this syndrome, which are motor speech disorder that is most often defined as apraxial speech, but there is definitely also dysarthria. And in the criteria, we put and or agrammatism in production and comprehension, because these two can occur. We can think of it as a bell curve in which there are a few cases in which they can present almost in isolation. So it's cases that are only agrammatic, and then there are cases that are only motor speech, but most cases really have both, or they evolve one into the other. And especially in the syndrome that is described as agrammatic, when it's isolated in grammar, is really not isolated to language. There is also a lot of executive disorders. So the definitions are never perfect. You know, human behavior is complex. So we need to think about some general templates that help us and guide us in our clinical practice, and then really study the exceptions and the most rare cases to make us evolve in the field. So this syndrome of non-fluent progressive aphasia that presents with these two syndromes, and I'll show you a couple of videos, is associated with left frontoinsular and subcortical network atrophy. And really we also like to define this syndrome, but what is spared? There is definitely spared semantics and spared comprehension of single words. And we'll go through that, but this is a very important point. The non-fluent progressive aphasia syndrome is a deficit in the language at the sentence level, not at the single word level. The single word level is much more in the semantic dementia patients. So it is a problem of comprehension, but a very different type of comprehension in the two disorders. The atrophy in this case that I'm showing you here is quite, and I usually use the neurological convention, so left is left and right is right, is in the posterior left frontal region. Usually the epicenter is in the opercular part of Broca's aphasia. You can see the atrophy here by enlarged sulcus, and really the striking preservation of the ventral language system. So it's a disorder that really follows most modern models of language, in which the ventral pathway is involved in comprehension of single words, and the dorsal part is involved in production, in speech production and understanding of grammar. So here's an example. This is a patient that shows both agrammatism and motor speech disorders. As I said, there are cases in which you see the extreme. For those of you in the audience who are used to stroke aphasia, we can think about it as a mild Broca's aphasia. Sometimes in spontaneous speech is not that evident. These are slow progressive disorders, and patients learn to work around them. So they might not make striking grammatical errors because they learn to produce simple sentences. Now, this might be different in French. I've learned that it's definitely different in Italian. And although I'm Italian, I've seen many more English-speaking patients, and I was surprised to see in my visits and collaborations in Italy how patients, even with a mild left frontal atrophy, could have striking grammatical deficits that I didn't see in my patients with the same atrophy in the US, who would have more apraxia speech. And that came the idea of, wait a minute, the different languages weight different functions of the brain differently, and that might influence how the disorder presents. And I've been thinking about this, stimulated by this conference, and also with my collaboration with a group in Paris, and also by a fellow that worked with us last year, Amandine, that some of you know. And it might be more difficult in French to distinguish the logopenic from the non-fluent variant because of the complexity of the language, but it's something that we could think about together. Anyway, this is an example of our non-fluent patients, and I hope the audio works.
Oh, man,
does it work?
A woman have a picnicky. A boy flies a kite, and next to the boy is a dog. Maybe they are family. A sailboat on the lake, and fishermen on a pier, and young man playing in the water.
So as you can hear, is a pretty mild case. She's describing this picture, and she has some symptoms of apraxia speech, hesitation, effortful. You can see that it takes her some work to produce language, and some distortions, which are typical of apraxia speech, and some grammatical errors typical of agrammatism. So dropping articles and pronouns, hesitations on using plurals and singulars. So we can think of grammar as the, and I need to apologize to the linguists in the audience, but as neurologists, we can think of grammar as the way that we put words together in sentences. So to put single words together in sentences, we need articles and pronouns, and we need to modify words and verbs to make them fit, whether we're talking in present or past. And so it's really how we construct sentences that make sense, and we need to understand who does what to whom. And she makes a subtle errors. So she's a typical example of a non-fluent progressive aphasia that has both grammatical and motor speech difficulties. So what is the network involved in non-fluent progressive aphasia? We said that the epicenter of the disorder is usually most commonly in the inferior frontal cortex, posterior, an area that is involved a lot in coordination of speech. In healthy controls, when we seed in this area, so we look at this area that we identified on the patients and look in healthy controls, how it is connected across the brain, we see atrophy also in the basal ganglia and in an area that is very important to program motor output, which is supplementary motor area and pre-supplementary motor area, so ventral cortex. And when we define with tractography what are the fibers connecting these networks, we identify this pathway that is very important, we call the Aslan pathway, that probably is the region where the disease will spread. It would start here in our mildest cases, they only have inferior frontal atrophy, and then it will spread across this network, first to the insula and basal ganglia and to supplementary motor area. And so the prediction would be that this disorder will start as a motor speech and grammatical deficit and might become, unfortunately, a more widespread motor disorder. And this network is, we know, is susceptible to tau pathology in the frontotemporal family of disorders. And so this would have been the hypothesis that this disorder is really caused by a tauopathy in the brain. So this is kind of a complicated experiment, and I will just tell you that this is the experiment that we did. We isolated the networks in the healthy controls, and when we have longitudinal data in the patients, and we matched the two to really go at the hypothesis that in this disorder, the atrophy and the clinical syndrome then will progress in this network. And I'm going to skip this. And this is what we saw. So this is longitudinal changes in the non-fluent progressive aphasia. And these are the network mapped in healthy controls. And when we look at the correlations of the two, we can see that there is longitudinal changes in the network that are most heavily connected. So really, this was a way to in vivo provide evidence for the transsynaptic spread of disease in the patients. And not only functional connectivity was related to progression of disease, but also structural connectivity. So within these frontal networks, there is higher progression of atrophy of the patients. We're lucky at UCSF to have a brain bank, and we follow our patients in research and in clinic, and they are very generous to donate their brain to us. It's a brain that I always show, and was my first patients at the Memory and Aging Center that I followed for 10 years. And it is quite an experience to have the opportunity to observe the brain of a patient that you followed for such a long time. You can see in the macro anatomy, the atrophy of this region that we see. This is the brain of the same patient that we follow through the year. This was the atrophy at year 1 when we saw her first, and it was very localized to the posterior part of Broca's area, which is this area here. And you can strikingly see it also in the pathology, how this gyrus here is much more atrophy than this one. It's much thinner. But you also can see that there is a dip here in the frontal lobe that was probably caused by atrophy in these medial frontal regions. And this is the evolution over time. So the atrophy progressed in the frontal lobe and in the parietal, more posterior region later on. This was her brain at year 1. And when we looked under the microscope of what pathology was involved, it was a tauopathy, and it's specifically cortical basal degeneration. In other cases, the pathology can be progressive supranuclear palsy. And unfortunately, over the years, most of these patients develop a more generalized motor syndrome. We believe that cases who have PSP, they're very difficult to distinguish. Progressive supranuclear palsy and cortical basal degeneration overlap both clinically and pathologically. So it's a difficult differential diagnosis. In our cases, these patients don't have more severe apraxia speech, but they have more severe dysarthria. So I'll show you and listen to an example.
In the foreground, there's a man and a woman on a blanket with a picnic basket. The man has glasses and is being
very difficult to coordinate speech with phonation. It's more of a steady disorder. So one of the main differences that Nina Dronkers taught me in my early years at the MAC in the distinguishing dysarthria from apraxia of speech is that in apraxia of speech, the errors are not, they're not continuous. It's not a disorder of primary motor function. It's a disorder of planning and motor responses. So it is not a constant deficit. It's not a difficulty with strength or with extrapyramidal functions. It's really a difficulty in planning speech, and we need to elicit errors by having patients repeat words with multisyllabic long words, multisyllabic words, and with consonant clusters. So we need to have, basically we need to have the tongue do this. This is what we do at the bedside to look at motor function in the hand. If you think about it, the tongue has the motor act of the tongue is much more difficult. It's the same muscle, and we need to have it move in the mouth back and forth to produce different sounds. And so when the tongue has difficulty being coordinated, especially with movements of the pharynx and larynx, then you have apraxia of speech. So if you think of a word and you can try to say it to yourself like artillery, you can see that basically the tongue is doing this in the mouth. So by having a patient repeat the word artillery many times or impossibility, you basically have the tongue do this and you elicit errors. If a patient is just saying something like impossibility, the movement stays at the tip of the tongue and is not that difficult to articulate. So depending on what the patient is saying in apraxia of speech, you can have errors or not. While in a dysarthria, it doesn't really matter what the patient is saying. It's always difficult. So this is an example of the pathology in this case that was progressive supranuchal palsy, and is an astonishing example of the spreading of tau in the white matter. You can really almost see it in the fibers, and this is a higher resolution. So, you know, we've seen the spreading, the involvement of white matter in this disorder that is a tauopathy, and this is the Aslant tract. So the tract that I was showing you before, this one. So we basically see microscopically what we see in the imaging of this spreading of the disorder within that tract. Okay, so we switch gear now to a completely different disorder, although it does present early on with language symptoms and anomia, and this is semantic dementia or semantic variant PPA. If we want to put it in the realm of progressive aphasia, we call it semantic variant PPA. We'll see that we're becoming more and more aware of the fact that this is not just a left hemisphere disorder, but also a right hemisphere. In the early description of semantic dementia, atrophy in the anterior temporal lobe was bilateral, but more left than right. And that was because we were observing patients that present with a language problem. But over the years, we realized that there are patients that really present more with a behavioral problem that we think is actually a semantic memory impairment for social cognition, and they present more with an isolated right temporal. And now we're a little bit in a conundrum of what to call those patients, because they don't really present with an aphasia, they present more with the behavioral symptom and maybe semantic memory deficit for faces and people. And so they don't really fit a PPA, aphasia syndrome. And so maybe the name semantic dementia would work better for them. But I really hate that term, because these patients early on are really not demented, and telling them that they have semantic dementia is something that I'm always a little bit resistant to. So I'm sure we'll find some other nomenclature that will work better. This is a really fascinating syndrome, is the one that I think teaches us more about cognition. It has taught a lot of to the community about semantic memory and cognition. And this is because this area of the brain, these anterior temporal lobes, are not often hit by stroke. They have a double vasculature. It's very rare to have selective, like isolated focal stroke in this area of the brain. So it was really ignored in the classic language model that we were talking about at the beginning of the talk in perisylvian cortex. This is definitely not perisylvian cortex, but it is a fundamental region for language processing, and studying these diseases really put it on the realm of the language networks. Now, I always get the question, what about epilepsy? And what about resection of the temporal lobes in epilepsy? I think that's very interesting. It has been studied, maybe not to the same detail as it has been studied in semantic degenerative disorders. Patients who undergo lobectomy because of seizures have a developmental disorders, and language is probably organized differently. So in that case, it presents a little bit differently. So I think that we learned a lot about what this area and network of the brain does from these disorders. So this is a typical scan that we would see of classic semantic dementia, atrophy of the anterior temporal lobe. Initially, neuroradiologists would read this as maybe old trauma or a cyst in the brain. I think now the field has evolved, but I wouldn't be surprised that sometimes it still doesn't get called in general neuroradiology. We still see it here, fortunately rarely now. So atrophy of the anterior temporal lobe, medial and lateral, but it's really visible. The temporal horn is enlarged, but you can also see that there is atrophy in the right as well, but much more on the left. When this is the anatomical syndrome, the typical presentation is cognitively is a selective loss of conceptual knowledge. Clinically, it presents as a severe naming, confrontational naming trouble. So patients have trouble naming objects and pictures of objects, difficulty understanding single words. Actually, if these patients understand single words, their understanding of grammar, what we were talking about before, is astonishingly good. They have trouble recognizing, identifying objects, people, and sounds. This has to be, again, investigated in a particular way because it's very sensitive to familiarity. So these patients can always identify objects that are very common in everyday life, glasses, watch. They might not be able to name it, but they will recognize it. If the items are a little bit less frequent and less prototypical, so maybe an eraser or a paper clip or some objects, and we'll see examples that are not, that we don't use really in our everyday life, then they will also have trouble identifying it. This was a bit of the confusion between a fluent aphasia and a semantic memory early on when we were talking about the early descriptions of PPA and the semantic dementia at the same time in Europe and in the US. I think in the US, the aphasia, the classic stroke aphasia batteries were used. And so patients will, so, you know, in those batteries, we have pencil and watch, and patients will be able to recognize the objects. And so some clinicians would say, well, semantic memory is spared. But actually, if we used more low frequency items, it wouldn't have been spared. They have a very interesting difficulty in reading in English and in French, I believe. They have difficulty reading irregular, atypically spelled words. This is a deficit that really doesn't appear in Italian or Spanish because they're highly regular languages. And we'll see, I'll show you an example. So the spelling of the words is predictable. The letter A in Italian is always A. It doesn't matter what letter follows or where it's put in the word, it's always the same. So if you learn the rule that the letter A is associated with A, you can read 90% of the words in Italian. And so Italian patients don't really show these symptoms, but I would believe that French patients would. Because of the network anatomy of the anterior temporal lobe, later symptoms involve behavioral changes. So in the non-fluent variant, later symptoms are usually a generalized motor syndrome. Here, patients from aphasic become quite behavioral as the disease goes on. Striking sparing of phonology and grammar, and this is very different from a classic Wernicke's aphasia. And when I worked on these patients with aphasiologists who were used to a stroke aphasia, they were definitely very surprised. And we talked about the anatomy. So here's an example. It's the errors that these patients make in naming is quite typical. So we said that it's very influenced by familiarity. So we would expect that this is an easier item to name, not to name, but to recognize than these two. The errors that these patients make is usually, since the semantic memory, and maybe I should say a word about what semantic memory is in contrast with episodic memory, as maybe some of you might not be familiar with this concept. So semantic memory is the part of long-term memory that is more encyclopedic, is knowing that a rabbit is a rabbit and that an apple is an apple. We don't know when we learned it. It's not linked to a specific time or a specific autobiographical experience. So we don't know when we learn that an apple is an apple, and we don't have a particular, in general, we don't have a particular autobiographic of emotional response to an apple. Very different will be remembering an episode like a wedding day or a graduation day, in which we have a very personal experience with and is linked to a specific time and place in someone's life. So we can think of semantic memory really as the encyclopedia, the Platonic idea of an object that is kind of detached from its physical attributes. So if this network of features that make an apple, what it tastes like, what color it is, where do we find it, how do we use it, does it live, does it not live, starts to degenerate, to disintegrate, we will keep the general kind of idea of it. So the type of errors that these patients typically make in naming is that they give us general terms. They tell us whether it's a fruit or whether it's a living thing, or sometimes where you can find it. But they won't call, they won't call never really an apple a pineapple. So they will not use a less frequent name for an object that they cannot name or recognize. So the typical error is that when they're faced to something that they recognize, but they don't know the name, they might give us a general term because they still have some kind of idea. It's not an all or nothing. So they might still say it's a fruit or it's an animal, or they might give us the prototypical item of that category. So you can think that if we start to lose the features that are typical of the rabbit, but we know it's an animal, we might give what for us is the prototypical animal. So we might call it dog or cat. Does that make sense at all? It's a lot of information, but so we can see an example. When will they show us that they really have a deficit in semantic memory is when we hit them with something that is low frequency. So it's really hard to get from context what this is. And actually right now, I can't even remember the name in English, a sled, okay? So it's a very atypical, non-frequent item. So this is the kind of item that they might say, I don't know what that is. Okay, I just don't know what the name is, but I also have no idea what that item is. So it's not just a problem of language, it's really a problem with semantic memory of that object. And here you see the objects that are used in a typical aphasia battery. As you can see, they're mainly high frequency. I mean, in this case, she'll have trouble with the word and the concept because she's quite advanced. But when we give these patients these items, they might show that they know what the items are. And that doesn't mean that they don't have a semantic memory problem. The only item they usually miss is the matches that are now low frequency. So here she is.
Vegetable, fruit. Dog.
Should I start again? The volume was low.
Is that a vegetable, fruit? Dog. I don't know what that is. Can.
It's a frog.
Cat. Fruit. Yeah, I know what that is. It's a, I forget what you call it.
Okay.
That's a dog.
So you hear how fluent she is in giving her responses. She has no hesitation. She produces sentences fine, but really has this striking naming deficit. I mean, if usually in the typical Boston naming test that we use in clinical setting, out of 64 items, they would name correctly, 7, 8, something like that, the most frequent. And then they would have these paraphasias for most of them in the context of spared phonology and sparse syntax. And you'll hear more here. And they have this kind of behavior that I've seen in semantic patients all over the world of like being surprised of not knowing. They have this sense that they should know. It's like, kind of really a disconnect between a familiarity with the word, with words in general, with language, but don't know what they're attached to. So I should know this word. Sentences, octopus, when you give them words and ask them to point to pictures, they really have this surprise.
The cup. A cup?
That's right.
Okay.
How about the matches?
Matches.
Well done.
This one?
Which one is the matches?
I don't know what the matches are. My brain is asleep. These are flowers. I know that.
Show me the pencil.
The pencil?
Yeah.
Is that this one?
Good. Show me the flower.
Right here. I knew the flower.
And the comb.
The comb. This one?
Good.
Okay. Again, you see here a slight behavioral change. She's struggling a lot and she's still making kind of little jokes with her voice. I think this is the beginning of this behavioral syndrome that might appear later on. In patients with stroke aphasia that would have this severe word comprehension deficit, they might have a Wernicke's aphasia, posterior temporal lesion. They would have much more trouble repeating and putting words into sentences than these patients have. So very typical, prototypical syndrome. So if we think about our network connectivity model, what does the anterior temporal lobe connect to? Mainly, there are mainly two pathways, one that goes posteriorly, the inferior longitudinal fasciculus, and one that goes anteriorly to orbitofrontal cortex, which we can think of as a big hub for behavior. And so we would predict that over time, these patients will get more and more aphasic, more and more difficulty with comprehension, and then we'll have difficulty with behavior. And this is what we see with these patients over time with imaging and clinically. Now, I anticipated that as time went by, and this has been really fascinated, it's been incredible really in these 20 years to see how the field evolved. There is really a right temporal variant of this disorder in which patients, instead of having more atrophy on the right temporal lobe, on the left temporal, excuse me, in the left temporal lobe, they have more atrophy on the right temporal lobe. And the head of our unit of the Memory and Aging Center, Bruce Miller, was one of the first to describe this syndrome. Again, there was a little bit of a cultural bias in which in Europe, especially in Italy, there were a description of this case, and it was this syndrome, and it was described as a progressive loss, progressive prosopagnosia, so progressive difficulty with recognizing faces. When we use, I don't like the term agnosia for semantic memory, because when we use the term agnosia, it's really usually limited to one modality of input, so either visual or auditory or tactile. And actually in this patients is not an agnosia, is a semantic memory deficit for emotions. But these patients, we didn't see them much. Bruce saw them because he was also working in psychiatry. And this is because these patients most often present as having loss of empathy. So loss of understanding, and let's use the word understanding to link it back to semantics, loss of understanding of emotions. So they don't understand, the symptom is that sometimes they tell you that they don't recognize faces, and they don't recognize not just faces, but the emotional trait of that person. So they lose even sense of familiarity, in the sense that they just cannot recognize a person for how they look physically, but also in what their personality is, and when they're fearful, and when they're angry, and when they're sad. So these patients go to marriage counseling. They don't come to a neurologist, because spouses start saying, my husband, my wife, they don't care about me anymore. The other day I cut my hand and I was bleeding all over the place, and she kept cooking, or he kept watching his football match. I'll always remember my first patients with this variant. The wife was so distraught. She was saying, he was the most caring, sweet man. And the other day we were on a canoe on the Russian River near here, and our daughter fell in the water, and he didn't show any sign of distress. He didn't care. She might have drowned, and he didn't care. But the issue was that it was not that he didn't care. He didn't understand. He didn't understand that she was fearful. He didn't understand the valence of that situation anymore. When did these patients come to a neurologist? When the atrophy spread to the left hemisphere and they became aphasic, because then it's clear that there's a neurological problem, and we associate it with the left hemisphere. That's what I was saying in the realm of behavior and emotions. We're not quite used to linking a specific lesion in a network with the symptom. Yes, disinhibition, being inappropriate, but not clearly phenotyping what is happening. So in these cases that we're starting to study, there are papers coming out quite regularly now. I'm really excited about that. This was like the first observation in the early 2000s that people that present with right more than left atrophy have trouble recognizing facial expressions. So if you ask them to match the name of an emotion or two faces with the same emotion, they couldn't say that this face is neutral or sad and this face is angry. They also don't know who the person is. So they do have this syndrome of prosopagnosia, which is not really an agnosia. They don't know who people are. And what we think the link is that the right temporal lobe is specialized for semantic memory for socially and emotionally relevant items. The disease is really the same. This is the work of Valentina Borghesani, who is now in Canada. And she really showed that in our path confirmed cases, this is a difficult diagnosis, especially retrospectively. So it is a little bit confusing now because some papers are coming out using retrospective data and retrospective descriptions of these patients. But back then, 15 years ago, we didn't know what to ask these patients. And so we might have described this as a loss of memory, but it wasn't really a loss of memory. And so I think it will take a little while to define the syndrome, but it is the same disease, whether it's right temporal or left temporal, if the patients are classified and examined with the tools that we need to identify that problem, then it is the same disease caused by TDP43C pathology. Not 100%, 85% of the cases, many of these cases also have Pick's disease. We like to call it semantic variant FTD. I think this would be a good name for it. Unfortunately, the term FTD is not the same, is not describing a syndrome. We don't have a term like aphasia for behavior. But I think this will give an idea that it is anterior temporal because it is semantic, but it is more of a behavioral syndrome. My colleagues here, we're still working on this. My colleagues who concentrate more on emotions and behavior might convince me to put an emotional here, like semantic emotional variant FTD, to make it even more complicated. I don't know, we'll find some name. I think it's important to distinguish these patients from the behavioral variant FTD, and I'm sorry I'm getting a little bit specialized here, because it's easier to predict pathology here. It's important to identify these patients, just like we identified semantic variant PPA in the left cases. We need to identify semantic variant FTD because these are the patients we want to send to trials. We have a trial starting this summer for TDP specific pathology, and we want to send these patients to these trials. Okay, briefly, how am I doing? Oh, super late. Briefly, phonology, logopenic progressive aphasia, posterior temporal parietal atrophy. Is that okay, or should I end here? You guys tell me. I'm sorry.
We have finished our day, you know, so you have all the time you need. There is no one.
Okay, and whoever needs to leave can leave without being rude. So it's I'm sorry, it's getting harder and harder to give a talk on PPA because they're really three different diseases, but I'll go through quickly. So patient with temporal parietal atrophy, so different from the frontal and anterior temporal that we saw, sometimes even more difficult to see in the scan. So beware of scans that are red and negative because they can have milder atrophy. They have less white matter involvement. So there is less of that lobar focal atrophy that you see in the other two syndromes. It's difficult to distinguish from the non-fluent variant because a deficit in phonological processing causes errors in speech. And so this was a big thing at the beginning of and it still is debated. It's difficult to distinguish these patients from the non-fluent cases, but it's very important because these cases have Alzheimer's disease and they should go into trials of Alzheimer's disease. I shouldn't say they have Alzheimer's disease. Marcel would get really mad at me. We're really good friends. He's been a mentor for me all through this. but we have very good discussions with Marcel Mesulam, and they don't 100% have Alzheimer's disease. There are some cases who have progranulin mutations, and so FTD that present with a syndrome that is similar to this. But they have, and you are actually in France, pioneers with Isabelle Lebert to study the progranulin. But 90% of the cases, they have Alzheimer's disease, and they're young, they're in their 50s and early 60s. They should go into trials for Alzheimer's disease. So they make phonological errors. We'll see examples of it. They have impaired repetition of sentences that, again, we said in non-fluent patients to elicit orders, you had to use multisyllabic, grammatically complex sentences. In semantic dementia, to elicit errors, you have to use non-familiar items. In the logopenic variant, to elicit differential errors, you have to have them hold on to phonological, so sounds of words and sentences, and for a long period of time. So what we call phonological short-term memory or phonological loop, which is, put simply, the mechanism that we use to remember a new phone number. If I give you a new list, a new phone number, to remember it, you have to rehearse it in your mind. You have to repeat it to yourself. You have to do this loop between hearing and speaking. And that's also when we're studying a new topic. You know, we use this kind of rehearsal to then move concepts to long-term memory. This is short-term memory, a few seconds. So these are patients that even forget what they're saying. They lose track when they talk. They learn to speak in short sentences as well that are grammatical. They usually don't make grammatical errors, but we need to speak in chunks to them. Otherwise, they lose the trace. It's and you hear examples. So when we construct sentences, so a typical task in general physiology that we use to test the phonological loop is to ask people to repeat series of numbers or sentences. But if the sentences are too common, for instance, today's a nice day, I don't really need to rehearse it. It's such a common sentence that if I hear it, I can repeat it. The telephone is ringing. Someone is at the door. If I ask to repeat these sentences, we're not really taxing phonological short-term memory. If I say Arthur was an oozy, oily snake, to repeat it, you really have to repeat it to yourself because it's not, it's infrequent. It's almost like asking separate words. So spared motor speech, although it might be difficult, you need really, it was really helpful for me to work with speech pathologists. And it's still sometimes I have trouble and I need to ask their help. I think we need speech pathologists or I guess logopedist, you would say in French, to help us with this. Their ears is trained to hear the differences. We're using a lot of automatic speech analysis now. I think the new tools of artificial intelligence and automated speech analysis will help us when we don't have a speech pathologist available. So here's an example. And you see how young this patient is. And so how important it is to make a correct diagnosis.
Bed.
Bed.
Nose.
Nose.
Pipe.
Pipe.
Window.
Window.
Banana.
Banana.
Snowball.
Snowball.
The telephone is ringing.
The telephone is ringing.
He is not coming back.
He is not coming back.
The pastry cook was elated.
The pastry cook was elated.
Okay, good.
So he lost the sound of that word that is less frequent in that context. And so he's making an error. It's not effortful though, like the first lady we heard with the non-fluent aphasia. He's just having trouble finding the sounds, knowing the sounds, not pronouncing them. But that can be hard to distinguish. So phonological errors versus motor speech errors. Phonology in French is so complex that I wonder if it's even more difficult in French than in English. I might be wrong, but that's, I wondered.
Bed.
Oh, sorry.
Bed.
No.
So very important is length, both of sentences and single words. So the longer the sentences, the hardest it is to repeat them. If they also less meaningful, then it's very difficult. Single words that are short, bed, door, table, no problem. So again, it's hard to just use a repetition score to classify these patients. It is a syndrome. We need to look at it all together. What is the neural network? This is work by Slađana Lukic, a postdoc in the lab. She correlated cortical thickness with the repetition in these patients and really saw that the association with this area in the superior posterior temporal lobe and in the supramarginal and angular gyri, very typically associated with repetition, especially of long phrases. So we said it's difficult to distinguish the two errors. Let's hear them back to back and see if we can distinguish the problems. So this is a severe logopenic patient. Actually, she's severe to start with because she's dyslexic, and we'd get to that in a moment. So she's not demented, she's very severely phonologically impaired.
Catastrophe. Okay.
Repeating the word catastrophe 5 times.
Tell me again what the
Catastrophe.
okay. Okay. Catastrophe.
Secrecy.
Microscopic.
Microscopic and microscopic and microscopic, microscopic, yeah, microscopic.
All right, you just have 2.
So not easy, huh? This lady has, the syllables are not distorted. She's really losing the sound of the word. And this woman instead knows the word, she knows the sounds, but she can't pronounce them. So it can be subtle, and but it has a very different anatomy and a very different biological basis.
Catastrophe.
So why do these patients with Alzheimer's disease have tau, not amyloid? Amyloid is all over the brain, and we have these PET scans now that can tell us in vivo that these patients have Alzheimer's disease. They have the proteins in the brain that cause Alzheimer's disease. The typical proteins are 2, amyloid or tau. Amyloid is all over the brain, so we don't think it's associated with the symptoms. Tau actually deposits in the areas of the brain that are most affected, so we think is more related. In these patients, it's very left hemisphere and very focal to this area of the brain for a long time. Why is that? And I'm going to skip this. These are the PET data that shows that the logopenic have Alzheimer's disease, and that atrophy progresses through this network. But what was interesting to me is when I started to study dyslexia, I mean, we had observed with Bruce for a long time that some of these patients, especially the very young ones, like the man you saw repeating, were telling us they were dyslexic. And not many people of that generation was diagnosed with dyslexia. Actually, they might not say that they were dyslexic. They will say that they had to repeat a class in school, or they're terrible spellers, which both in the US and in France is a big deal, not being able to spell. I know because my dyslexic daughter was going to a French school, and it was a big deal that she couldn't do the dictées. And so I started looking at this and realizing that, you know, the most common form of dyslexia is a phonological disorders. And then when we look at functional imaging studies in dyslexic readers, the area that it has more difficulty activating is this area in the posterior temporal parietal region that really corresponds to the atrophy of the logopedic progressive aphasia. So there is a network overlap. So could it be that logopenic PPA is just Alzheimer's disease in a dyslexic individual, or in an individual in a family of dyslexics, because we know that neurodevelopmental disorder are familiar. So can this be one of the factors of susceptibility? Does that mean that dyslexia causes Alzheimer's disease? No, we don't know if there is a biological overlap, but it could be that it influences the phenotype of the disorder. It can be both things. In some cases, there might be a biological overlap. Zach Miller's work in our lab now is quite, this is the first description, showed that actually the patients, you know, there were description of neurodevelopmental frequency, high frequency of neurodevelopmental disorders in PPA. His work showed that is really network specific at least sometimes. So the logopenic variant, even in this retrospective study in which we didn't ask for neurodevelopmental differences, he saw a higher frequency of dyslexia in LV-PPA in the logopenic variant compared to the other PPA variants. It has a longer disease course than the other PPAs. In this case, who was again our first patient who had all the had an MRI, you see how subtle it is. She knew she was dyslexic. She's the patient, and this is something that, again, to the young researchers or clinician here, the observation of a single patient is really fundamental for us. She was the first logopenic patient that Nina and I saw together and described really the syndrome based on her. Very subtle atrophy, very subtle in the posterior superior temporal gyrus is inferior parietal region. It was more visible in FDG PET the following year, and she had a positive PIB scan. Very hard to think that this woman had Alzheimer's disease, right? Like if you see her, she doesn't look like the Alzheimer's disease patient that we would present. She was a teacher, so she knew she was dyslexic, that her brother was dyslexic. And in the cases that they tell us that they have a neurodevelopmental difference, it seems like the aphasia, only the isolated aphasia part of the disorder is longer than 2 years. So I think I will stop here. There was a bit of a discussion on what dyslexia is, but I already kept you too long. So the general idea, I'm going to thank the Memory and Aging Center and the Language and Neurobiology Lab. It's been an incredible adventure to jump around continents and learn from different people who have different views. One of the lessons that I learned is to really be open-minded about other ideas and think in context. And you know, the lesson that we learned is when we see adult patients, we want to ask the pediatric history, and when we see pediatric patients, we want to ask about their grandparents. That the way the brain was built influences how the brain ages. And yes, I want to thank all my colleagues. It's been really an amazing adventure. Thank you. And someone is ringing at the door. I don't know who it is. I'm going to let it be. I don't know if anybody still has the strength for any questions. My apologies for going so long.
Thank you. Please do not apologize. It was an outstanding conference, and it was very, very nice for us to hear it after 2 days of lectures on PPA.
Oh, wow.
And other focal syndromes. It was only half a day actually about PPA specifically. And it was great to see some English examples as well. So we were 75 to follow you, Marilu, and for the next following minutes, if you allow us a little bit more of your time, I will see if questions are coming. The panelists can intervene as they wish, and the students can ask their questions through the chat, but I will read them for you, of course. Maybe I have a first question. You said, oh, sorry. There are many questions. Well, I will shut up. So maybe first Olivier. I saw.
No, it was Mathieu first.
It was Mathieu first.
Olivier first, please.
Oh, okay. Thank you for this very nice talk. I'm happy to see that American patients are quite similar to French patients and have the same impairments. We're not very comfortable with the term fluent or non-fluent aphasia in France. And what would you call fluent and non-fluent?
Oh, I agree. I'm not either. You know, I'm not either, because especially because it was a term that was again adopted in stroke aphasia, and it doesn't really apply to these patients. Who's fluent and non-fluent, and patients can be more or less fluent depending on where they are. So I agree with you that it is an imperfect term, and it was kind of inherited, and I would be happy not to use it, but it's hard to let go of it. So fluent and non-fluent is almost a kind of a clinical impression right now, you know, how much they speak and how hard it is for them to speak. It's very clear that semantic patients are fluent. They're sometimes hyperfluent, but what we mean by non-fluent in the logopenic versus the other syndromes, it's hard. So we could decide to completely divide agrammatic and motor speech disorder in the larger non-fluent category. But then what do we do with the patients who have both? I mean, it's the nomenclature of this. I'm sure there'll be a new phase. Yeah, go ahead.
Would you prefer effortful versus non-effortful?
I'm sorry.
Would you prefer effortful versus non-effortful?
Yeah, I would like effortful. It's a difficult term to use. Yes, it's the patients with apraxia speech will have a lot of trouble using it. But yes, I like that, effortful and non-effortful. But in some ways, yeah, it's motorically effortful, right? Because it's phonologically effortful too. I don't know. I try to be flexible, and as long as we know what we're talking about. That was kind of, I think before the criteria paper, we really didn't know exactly what we were talking, if we were talking about the same thing. I had a lot of trouble going back and forth between the different literature. I like the idea of effortful. My first reaction was, oh my God, how are the patients gonna say it? Because we have our nomenclature, and that's what we tell the patients. And it's such a difficult, right? Us clinicians that face the patients is hard to, the PPA term is easy for them. I have trouble with the PPA terms too. I think I was very surprised that John Hodges was willing to let go of the semantic dementia word for semantic variant PPA, but yeah. So I don't think there is, it's difficult to find the perfect term, let's put it this way. But I'm open to it. I'm not married to any term.
So I have now a gallery of all the panelists in front of me, so I won't miss any arm raised. I think there was Mathieu, and I will next introduce you to Sophia Yakomb.
Thank you, thank you for your very nice and very clear talk. I'm very interested in your works on connectivity, and I'm wondering if you had some data about contralateral consequence of left hemispheric lesions, especially in terms on hypo or hyper connectivity in the right hemisphere, and how would you interpret this consequence as in terms of compensation or in terms of a consequence about participation to the disturbance? For example, did you think that there's a role of right hemisphere contralateral consequence of left hemispheric lesions on prosody? For example, that's an example. So I'm interested in that field for that field by your answer.
Great questions. I actually see a question from Amandine Girardieu. I still have trouble pronouncing your name.
Amandine Girardieu. That's one of our students.
I don't know if you presented your prosody work, but we haven't quite made that link. It's a super interesting question. We do see hyperactivation contralaterally, and sometimes even in the same hemisphere. So for instance, in the semantic variant patients, we see hyperactivation in the dorsal network. In the non-fluent patients, we see a hyperactivation in the right frontal lobes. There has been some work showing that in Alzheimer's disease, MCI patients have hyperactivation is it an early sign of degeneration really and inflammation in those areas, or is it a tentative compensation? Our early data, we actively studying this, show that it's not related to better performance. But I'm not sure we're using the right tasks, right? Like we didn't study prosody enough to show that maybe the right temporal activation that we see is related to improve or decrease prosody. We didn't have, I mean, the fortune that we have at the Memory and Aging Center is that we have many investigators studying the same patients. And Virginia Sturm is interested in emotions, and Kate Rankin is interested in social cognition. So I might have social cognition data in some of my patients, but not that systematically. So the brief answer is yes, we are studying it. Yes, there is hyperactivation in initially spared networks. They seem to have a lot of tau in it in the Alzheimer's patients. So as much as I want to believe is for our patients that is compensation, I am afraid is early degeneration. So it's a sign of early network, degeneration, like network related degeneration, but I'm not sure yet. I hope it's not. I hope we can use it for compensation, but I'm not like using TMS or TDCS in those regions to help the patients, but I can't tell for sure yet.
Thank you, Marilu. Next question from Sophie Oriacom, and then we'll go to to Amandine's question, which is one of our students. Sophie Oriacom is the head of Bordeaux Memory Clinic.
Thank you so much for your talk. My question was about TDCS, actually. What about? Do you have any experience with this type of care for PPA people, and what is your opinion also on speech therapy?
Oh yes, so in speech therapy we have a very active research program in collaboration with Maya Henry in Texas. She was a postdoc with us and then moved to Texas. Speech therapy that is very, again, thought through based on cognitive domains that are spared and impaired in patients. Maya has shown that is extremely useful. I'm sure there is some useful communication strategies that work for everybody, but a cognitive-based speech therapy, Maya has shown that is very useful and it should be, I think, available to everybody. If we associate TDCS to it, will it improve? There is some data that shows that it will improve. We were about, before the pandemic hit, we were about to start a study in non-fluent patients. I really wanted to, interestingly, many of the TDCS studies out there have looked at functions that are, to me, not that immediately salient to the patients. So I really wanted to do a motor speech kind of TDCS with on the inferior frontal gyrus, and we didn't manage because of the pandemic. There are some encouraging data. TMS would probably be even more, but we have a very strict do no harm policy that we, these patients are suffering a lot, and doing TMS of the anterior temporal lobe, I wouldn't do it, even if a left frontal lobe. Maybe we're wrong. Maybe we should do it, but we've been, and also in the, we've shown, the literature has shown that the early onset Alzheimer's disease patients have a decreased threshold for seizures. We've debated it, but it hasn't, we haven't done it much.
Thank you.
So not huge experience, but definitely in the literature, there is some positive results that are encouraging.
I move to Amandine's question as planned. Thank you, Marilu, for this great talk. You explained that there are hypotheses for network vulnerability, like dyslexia for LVPPA. But are there hypotheses for the selective vulnerability to a specific toxic protein, for instance, TDPC in anterior temporal lobe?
So, I mean, the toxic protein is TDPC that accumulates. Why is it? I guess the question is, why is it TDPC in the anterior temporal lobe? And I think the biggest hypothesis right now is that is for some reason that's an area of the brain that is very susceptible to inflammation and autoimmunity. You know, we know the clinicians, neurologists in the audience know that many autoimmune disorders and paraneoplastic syndromes interest the anterior temporal lobes. And so I think that's the main hypothesis right now, that there might be some immunological, I'm not a basic scientist, I'm sure others in the lab will have a better answer for you, Amandine. But there is a link to inflammation for sure.
There is a question from Serge Belliard from Rennes.
Thank you for this great talk. My question is, do you think that in the future there will be only 3 subtypes of APP, or do you think that other subtypes will be described in the future? I think about, for example, some patients with phonological or syntactic comprehension deficits, or what has been called by Luria dynamic aphasia or motor progressive aphasia. What do you think about this motor transcortical aphasia? What do you think about these subtypes?
You know, I think they're definitely there. I've seen a couple of super interesting dynamic aphasia cases. I've been thinking of whether they are an aphasia variant of BVFTD, really, right? And they have a lot of medial frontal and anterior frontal atrophy. They really have this apathy for language. You know, if I put it in context with my behavioral colleagues that concentrate more on behavior, many of the BVFTD patients also have left frontal atrophy, much more than we expected, really. And I wonder whether some of the symptoms of BVFTD are really more aphasic inside, like patients are not speaking. They're not really generally apathetic. They have an apathy for language. So absolutely, yes. I'm always, you know, again, keeping my clinician hat on. I'm very conflicted on whether to make the syndrome more complicated. I think maybe we have to have 2 tiers, you know, tiers in which we scientists and use very specific names for some syndromes, and then use a more general term for the patients. But I totally see there is a left prefrontal syndrome. Is not left posterior frontal, is more left prefrontal. And if the anatomy is different, the clinical syndrome will be different. So yes, absolutely. And maybe as we get better distinguish them, we will find more patients, and we will need to describe a progressive dynamic aphasia. How it interacts with BVFTD is like, but yes, so absolutely, there are other variants and other presentations. And the more we know, the more consistent they will be. So I guess the answer is yes, there are definitely other variants. And as we know more, we'll define them better.
Thank you.
Thank you. Are there other questions? Yes, Florence Pasquier. Florence, you have to activate your microphone.
Yes, sorry. It's not a question, it's just a comment. You really, thank you very much again, and you really won your formal invitation, and I hope you will come to Lille.
Thank you, I count on it. Thank you for being there.
So there is another question from Mai Tran. Mai Tran is the head of the Faculty of speech therapy here in Lille, and she wrote something about your debate about this dichotomy between effortful and not effortful. She's asking yourselves, should it concern the motor level, the level of articulation, phonetic realization, or the language level, the construct sentence or discourse? Well, that's.
I absolutely, but try to get neurologists to do that. You know, it's again multi-disciplinary patients, researchers, neurologists, psychiatrists, speech pathologists. We all have different level of knowledge, different nomenclature, and at some point, I think to work together, we need to. My view is to be able to work together and having a common language, we need to be willing to be imperfect, but then really go deeply in describing what the patients have. I don't think you can get a neurologist at the bedside, especially a more general neurologist, not us specialists, to even pay attention to these syndromes if they're too difficult, if they're too precise. You know, construct sentence or discourse, a general neurologist who need to refer the patients to us, they won't be able to do it. I mean, but I totally agree with you, that is the issue. And we might add, I don't know how we would call it, but willingness to speak, to encompass the adynamic aphasia. It becomes difficult, but we need to somehow get there. Keep it simple enough so that patients can be identified and taken care of, but then also be precise enough to incorporate all the knowledge we have of linguistics and cognitive neuroscience. And I think that's the challenge. And there will always be someone unhappy that we weren't precise enough. I'm having that experience in the emotion work too. There's huge debates, you know, trying to define the right temporal syndrome. You know, there are people who think emotions are innate and people who think emotions are acquired through experience, and they have different terminology, they use different constructs. And just like in language, we have, you know, the Chomskyan and the non-Chomskyan, and it is a challenge, and I think it's a challenge that we need to face, to be able to be simple enough that we can take care of our patients and identify them, but then also make the field evolve by studying really in details the both molecular and cognitive underpinnings of their syndromes. So I'm sorry, I don't have a perfect answer. I hope I get the general idea.
Thank you very much, Marilu. I think that we will stop there. We will go home and have dinner while you will start your day. It's fantastic.
Finish my coffee.
Thank you so much.
Thank you so much. I think it's not an overstatement to say that we appreciated a lot your conference, your expertise, your kindness. So see you in Lille when this pandemic thing is over.
Next year. Thank you. Bye-bye.
Bye.
Good evening.
Goodbye. Thank you all.